All right, let's dive right into an absolute game changer in agricultural biotechnology. Today, we're looking at a major scientific breakthrough that honestly feels straight out of a science fiction novel, but it's happening right now in real-world labs. We're going to explore how scientists have figured out a way to make precise, targeted genetic upgrades to crops, specifically potatoes, without integrating a single piece of foreign DNA into the plant's genome. For plant breeding, this is huge. I want you to imagine something for a second. Imagine upgrading a crop's genetics to give it better yields, stronger disease resistance, or higher nutritional value, but doing it without leaving even a trace of foreign DNA behind. Is that actually possible? Well, historically, whenever we've modified a plant's genome, we've had to deliver those new instructions using DNA vectors. The problem is that almost always leaves some genetic baggage behind. But in this explainer, we're unpacking a peer-reviewed study that proves we can now perform these edits invisibly, cleanly, and completely transgene-free.
Okay, here's our roadmap for this explainer. First up, the potato breeding problem. Number 2, enter CRISPR-Cas9 tools. 3, the DNA-free innovation. 4, inside the protoplast workflow. 5, validating those transgene-free results. And finally, the future of farming. Let's start with Section 1, the potato breeding problem, and specifically, the challenge of tetraploids.
So the regular cultivated potato, Solanum tuberosum, is incredibly difficult to improve through conventional breeding. Traditional crossbreeding is notoriously slow and highly unpredictable. Why? Because the potato is tetraploid, meaning it has 4 full sets of chromosomes, and it's highly heterozygous. Because of this tetrasomic inheritance, if you try to crossbreed potatoes to introduce just one single beneficial trait, it's like shuffling a massive chaotic deck of genetic cards. Sure, you might get your new trait, but you'll almost certainly lose the specific characteristics that made that commercial variety great to begin with. The alternative? Genome editing. This allows scientists to essentially surgically add or alter targeted traits directly in an existing variety, completely bypassing that crossbreeding headache.
Moving to Section 2. Enter CRISPR-Cas9 tools. Let's talk precision genome editing.
You've likely heard the term before, but let's define it clearly based on our source material. Think of CRISPR-Cas9 as a pair of microscopic molecular scissors. It works by combining 2 key things. First, a guide RNA, which basically acts like a GPS system programmed to find a very specific sequence of DNA. Second, a Cas9 endonuclease, which is the actual cutting tool. Together, they zero in on the target region and create a highly precise double-stranded break right in the DNA. But here's the catch. There's historically been a major flaw in how this is done in plants. Standard genome editing often delivers those CRISPR instructions into the plant cell via plasmid DNA vectors. Essentially, scientists send foreign DNA into the cell to tell it how to build the Cas9 protein and guide RNA. The problem with that? When the cell repairs that double-stranded break we just talked about, it can accidentally grab fragments of that foreign plasmid DNA and stitch it right into its own potato genome. Unintended DNA insertions from plasmid DNA happen at a shockingly high frequency. And obviously, from both a commercial and regulatory perspective, having random foreign DNA integrated into your crop is exactly the opposite of what you want.
Which brings us to Section 3, the DNA-free innovation. Let's look at the RNP delivery method. To completely eliminate the risk of those unintended DNA inserts, researchers decided to just ditch the idea of sending DNA instruction manuals into the cell altogether. Instead, they used ribonucleoproteins, or RNAs. An RNA is a fully preassembled complex. It's basically a microscopic strike team, where the actual Cas9 protein is already bound to the guide RNA. Because it's fully built before it goes in, it doesn't require any transcription or translation by the cell. It enters and goes to work immediately. And the absolute best part? Because you're delivering a protein and RNA and no DNA whatsoever, there is literally no plasmid DNA present to accidentally get inserted into the plant's genome.
So what did they target? They sent this strike team after the potato GBSS gene. Well, specifically a region called GT4 inside exon 9 of that gene. Now, this gene is responsible for the synthesis of amylose starch. By knocking out its function, the researchers could definitively test if this RNP method worked. If successful, knocking it out would produce a potato with high amylopectin starch, which, by the way, is a highly valuable commercial trait.
Let's step into the lab for Section 4. Inside the protoplast workflow, where we edit naked plant cells. The secret to pulling this off lies in a fascinating state called protoplasts. Normally, plant cells have a rigid protective outer armor known as the cell wall.
But to get our crisp RNPs efficiently inside, scientists use special enzymes to temporarily strip away that armor. What you're left with are protoplasts. These are naked, highly vulnerable, but incredibly receptive plant cells.
Okay, let's see how this builds into an actual lab workflow. Step 1. Researchers isolate these naked protoplasts from a potato cultivar. Step 2. They introduce the preassembled CRISPR RNPs using a chemical called PEG, or polyethylene glycol. What PEG does is temporarily make the cell membrane permeable so the RNP's strike team can slip right inside. Step 3. The RNPs do their job, cutting the targeted GBSS gene, and then the scientists carefully coax those single, naked cells into regenerating back into entire, fully grown potato plants. And finally, step 4, they analyze the plants to validate the mutations and, crucially, check if they are totally free of foreign DNA.
On to Section 5, validating transgene-free potatoes. Let's check for those DNA inserts.
When the researchers compared 3 different delivery methods, the results were frankly stunning. Using standard plasma DNA, a massive 67% of the mutated lines ended up with unintended foreign DNA inserts. Next, they tried an RNP made with in vitro transcribed RNA, labeled IVT RNP here. That resulted in an 86 to 100% rate of unintended inserts. Why? Because the RNA production process left behind tiny microscopic DNA templates, and the plant just eagerly grabbed them and stitched them into its genome. But, and here is the massive breakthrough, when they used cRNP, where the guide RNA was produced synthetically in a 100% pure DNA-free environment, the rate of unintended DNA inserts was absolutely zero. Zero! Not a single regenerated shoot contained foreign DNA.
And it wasn't just clean, it was highly effective. They achieved an impressive 9% mutation rate using that synthetic cRNP method with a 40% PEG concentration. Out of those regenerated shoots, they successfully yielded a 2 to 3% complete knockout rate. That means all 4 copies of the targeted allele in this super complex tetraploid crop were knocked out perfectly, without a single trace of foreign DNA. To put that in perspective, for a commercial breeding program dealing with a tetraploid crop, a 2 to 3% success rate for a perfectly clean knockout is a phenomenal result.
Now, what's really fascinating about this next visual is that it's the actual physical ta-da moment of this invisible genetic edit. This is an iodine stain of starch granules taken from the tiny tubers of those regenerated plants. On the right, the wild-type potato starch stains a deep blue. That tells us the normal amylose starch is present. But on the left, the starch from our clean, DNA-free knockout potatoes stains a distinct red-brown color. This proves visually right before our eyes that the GBSS enzyme function was entirely eradicated, leaving pure amylopectin starch. The edit worked flawlessly. Let's wrap things up with Section 6, the future of farming and the global impact of this work. So, why go through all this effort to be entirely DNA-free? A huge driver here is the global regulatory landscape. Plants with foreign DNA inserts are often heavily regulated as genetically modified organisms, or GMOs, and the legislation around this, especially in places like the European Union, is incredibly complex. However, genome-edited plants that simply have small, precise inserts or deletions, exactly like the ones created by this clean CRISPR RNP method, are chemically indistinguishable from plants carrying naturally occurring mutations or mutations you'd get from traditional crossbreeding. Because they are 100% transgene-free, they have a real potential to bypass strict GMO legislation, which could drastically speed up the timeline from the laboratory right to the farm. The broader implications here for our global food supply are staggering. If we can cleanly edit crops, we can rapidly develop plants that are resilient against evolving diseases and extreme climate shifts, all while boosting their nutritional value. But we have to keep a balanced view here. The researchers are very upfront that significant hurdles remain. Taking a single, naked protoplast cell and regenerating an entire plant from it is notoriously tricky, which means regeneration efficiency is currently quite low. Also, while this workflow is fantastic for potatoes, it's pretty difficult right now to apply it across all crop species. So it's a massive leap forward, but the science is still actively evolving.
Which leaves us with a final thought to ponder today. If scientists can now reprogram the incredibly complex traits of our crops from the inside out, completely avoiding the chaotic genetic shuffling of traditional breeding, and doing it without leaving a single trace of foreign DNA behind, how will this invisible technology ultimately reshape the future of our global food supply? It is a truly fascinating frontier of science, and definitely one we'll be keeping a close eye on. Thanks for joining me for this explainer, and keep learning.